Dissolution is one of the critical attributes of the drug product, and it is always part of the specification. And the dissolution is always expressed in the form of the Q. For example, the specification for dissolution is not less than 75% Q or not less than 85% Q. So what this Q means and why this Q is generally used in the expressing the dissolution specification. And as a part of this video, we will try to understand the importance of the Q and how this Q can be used in designing the specification. So let us begin with the presentation. Let us try to understand the references for Q given in different pharmacopeials. So if you look into the Dissolution General Chapter in the USD, which is 711, you will find the definition of Q. This way, the quantity Q is the amount of dissolved active ingredient specified in the individual monograph, expressed as a percentage of the label content of the dosage unit. So Q means the amount of dissolved API during the dissolution process, and which is mostly expressed in terms of the percentage. That is the definition of Q according to the USP General Chapter 711. If you want to understand the definition of cube in the context with the AP General Chapter 2, 9.3 now. Here is the definition. The quantity Q is the specified amount of dissolved active substance expressed as a percentage of the label content. So, again, the EP talks about the percentage of the API dissolved in the dissolution process, and that is all about the Q. Now let us discuss about how this Q value can be calculated. See, you must have been saying the dissolution specification, not add only one step, but at the three different steps, S1, S2, S3, in case of immediate release dosage form, or L1, L2, L3 in case of the modified release dosage forms. So we will take one example, and according to the example, we will try to understand how this Q value at these several steps can be identified. And once you understand the value of the Q, you can also understand whether the product meets to the specification or not at any given particular state. Let us see USP resolution acceptance criteria for immediate release dosage form. And let us assume, for explaining this example, that the value for our Q is 85% now. If you say my Q is equal to 85%, then you have to express the specification not less than 85% Q, where your cube is 85%. Acceptance criteria for this product through three different steps will be as below. The first one is S1 state, where 6 units are generally tested. So what is the requirement for S1 step? All 6 unit results should be greater than Q plus 5%. Means none of the unit must have a dissolution result less than Q plus 5%. Now what is the Q in this example? It is 85%. So 85 plus 5 becomes 90%. So in case if all the units, if all the 6 units are at least equal to 90% are greater than 90%, then you can actually say my resolution meets the requirement at the S1 stage. But in case if 1 or 2 units are less than 90%, then you have to continue the dissolution at the S2 stage. So how S2 stage can be conducted at S2 stage? You have to conduct the dissolution on 6 additional units. See, you have conducted the dissolution with 6 units at S1 stage. And that is not the specific, that is not meeting the specification. And hence you are now moving with the S2 stage, where you are going to conduct dissolution on 6 more units. So in total, at the end of S2 stage, you are going to have 12 resolution unit data, 6 from S1 and 6 from S2. So what is the acceptance criteria at S2 level? An average of 12 unit. An average of 12 unit means 6 from stage 1 and 6 from stage 2 is equal to or greater than exactly Q value. What is Q value in our case? It is 85%. Look at here. Okay, so in case if the average, I'm not talking about the individual unit at S2 level, I'm talking about the average of 12 unit, should be greater than 85%, which is a Q value. In case if there are units which are less than 85%, there could be possibility that all not unit are equal to 85%. But if some units are less than 85%, it's going to be acceptable, provided no unit is less than Q minus 15%. No unit should be less than Q minus 15%. And what is the cube in our case? It is 85% minus 15% going to become 70%. So no unit should be less than 70%. And my average of this 12 units should be 85%. In that case, I can confirm and conclude that my product meets the dissolution specification at S2 stage. But for some reason, if I have a sum unit which is less than Q minus 15%, so for example 60%, then in that case I have to move with the S3 stage now. So what is going to happen at S3 stage? I am going to conduct dissolution on 12 extra units. So what is the acceptance criteria? So 12 additional unit has to be tested, as I talked, an average of 24 units. So how this 24 units data got collected? Because 6 units data from S1 stage at another 6 unit data from S2 stage, and additionally 12 unit data from the S3 stage. So altogether, we are now going to have a data for 24 dissolution units, S1 plus 2 plus S3. Now, what is the requirement? The average of all these 24 units data should not be or should be equal or greater than 85%. Means what should be greater than or equal to Q. So what is our Q in this example? It is 85%. So in case if I found that the average of 24 unit is at least 85% or greater than 85%, then I can say that my product meets the specification at S3 level. But there could be chart that some units could be higher and some unit could be low. So there is a requirement as far as the lower dissolution units are concerned. And what S2, S3 stage talks about, not more than 2 units are less than Q minus 15%. In case if I have some units, only 2 units up to 70%, then still I can say my product meets the S3 criteria, but provided the average should be equal to or greater than 85%. I hope you understand this point. An additional requirement is that no unit is less than Q minus 25%. So none of the units should be less than Q minus 25%. As our Q is 85%, so the 85 minus 25 becomes 60%. So in case if none of the unit is less than 60%, then I can certainly conclude product may take the specification at S3 level. So S3 acceptance criteria has 3 different requirements. The first point we talked about having the average greater than or equal to at least Q value. The second important criteria is for not more than 2 units less than Q minus 15%. And the third acceptance criteria is what none of the units should be less than Q minus 25%. So this is all about how to understand the different stages of the specification and how one can conclude whether the product meets the spec or not. Now the next point is very important, and I wanted to throw some light on this point as well. Why do we need to add 5% to the Q value at the S1 stage? So you must have seen that S1 stage, our specification for dissolution is not less than Q plus 5. So why this 5 has been added at the only initial S1 stage? Because if you go S to S3, we only talk about average equal to Q, and there is no requirement of Q plus 5 as the average or as an individual dissolution or unit of the dissolution. We do this because the specifications are all based on N equal to 12, but will analyze only 6 samples, which is an abbreviated testing at S1 stage. So if you look into the bioequivalent study, especially in the case of genetic drug products, we always conduct the study on 12 volunteers or the subjects. In case of comparing the dissolution results from reference and test, we always compare the dissolution data from 12 units. But in case of our routines testing or stability testing, we only conduct a dissolution on 6 unit, which can be called as abbreviated testing. We do not have a result dissolution data on its 12 units when starting with the smaller samples and equal to 6. We need tighter criteria. Since you don't have as much statistical confidence as we increase the sample data set, say N equal to 12, especially at S2 stage, we get closer to using the Q since you gain more confidence in your data. So to have the extra confidence because of the lack of statistical confidence at S1 or 11 stage unit. So let me know your thought on the entire discussion that we have in the particular video. And please don't forget to put your comment in the comment box below. Thank you so much.